until now, I was talking to you about DNA, mapping the genes and finding out what genes are altered. But we moved on. Remember, I told you earlier on that DNA makes RNA, RNA makes proteins. Proteins are the most, that's the business end of the unit. And mapping the proteins in large numbers has been a difficult task until recently. So there is a big, monumental National Cancer Institute, taxpayer funded research that's currently ongoing, mapping the proteins in large numbers. And we do find in some instances the DNA is not messed up, but the protein is present in way high numbers. And you don't have to know how to read this graph, but the important thing is this is all the proteins are clustered together, but the one is standing outside in a big way is a protein called SHIP2 that goes by the gene name PTP11. And this particular protein is really off the chart, present almost exclusively in EGFR mutant lung cancer patients. Why is this important? This is important because SHIP2 now we can go after with specific therapies. There are now combination trials that are currently ongoing with SHIP2 inhibitors in EGFR mutant patients. And the same way with KRAS mutations, there is a lot of interest in this targeting a protein called SOS1. The point of this slide is to show that now we are studying proteins using really state-of-the-art what we call mass spectrometry studies to really find out what proteins are altered and try to understand how different gene alterations lead to different kinds of protein alterations. And that's really opened up our knowledge in this area quite a bit. And all of them hopefully would help us understand, help us build more new drugs. Over time, I would say that these cancer cells are not static. They begin with EGFR mutation here, and what happens, if you give therapy, these mutations evolve over time. This may be hard to read, but this is a normal cell, and where you have this that had all the EGFR mutation became a cancer cell, and then over time, these their genome or the entire suite of genes became double and triple in number. And then what happens is that within that you have several groups of cells growing. It's almost like a gang, different gangs, different groups that grow with different gene alterations. One goes to the liver, one goes to the brain, one becomes tougher to treat. One responds very well to treatment. Two of them don't respond to treatment. That's why on treatment, you can have, let's say you have lung or liver metastasis, the lung lesions respond very well, but the liver lesions begin to grow. And that's why, we come up with different strategies and the cancer cells evolve over time. And now we are really doing something unique where we take a patient's primary tumor in the lung and then grab a sample of the tumor tissue from the brain and study them together and to see how are these cells changed from the lungs to the brain, what has happened. And this is a work that we did a while ago, and it's still a ongoing work. And part of it we have published already. It turns out that compared to the primary tumor, the metastatic tumor in the brain has decreased immune response and decreased inflammations. In other words, the immune cells are not there to kill the cancer cells. And these cells, they become more like a brain cells. The lung cells become a little bit more like a brain cells and they tend to grow happily there. And we have to understand why do they grow nicely in the brain? What can we do to prevent them from growing in the brain? The cancer cells get disseminated widely, perhaps very early on, even before the patients have symptoms, but they are controlled by the body's protective system in many places. But now and then the cancer cells happily grow in some places, and we need to find out what leads them to grow there and then try to target them. So we need to understand how these cells are altered in different groups, and that's how we can really go after them.